It is 12 o'clock Pacific, 1 Mountain. Getting everyone joining in today. Welcome. All right, hello and welcome to this month's Biomimicry Fireside Chat. I'm Lexa Moray, Communications Director at the Biomimicry Institute. Before we get started, I'll go over a few housekeeping items so you know how today's meetup will work. As I do this, let's go ahead and get a poll going to see what the group's challenges have been for solving problems. Let's see here. Okay, you should see a poll on your screen now. Please take a moment to respond and I'll close it in a few minutes. At any time during this webinar, you'll have the chance to submit your questions to our guest speakers. To do so, just type your question into that Q&A at the bottom of the control panel. We also captured a lot of your questions from registration. As time allows, we'll address as many as we can during and at the end of this conversation. And then for any of those topics that don't get explored, we'll follow up on social media and with articles on our blog, Asking Nature. And of course, we'll also continue to shape future fireside chats to focus specifically on these areas. During our last meetup, I heard from many of you that you wanted a way to connect with each other, share ideas, and collaborate. While we don't have the capability of sharing your videos during the Zoom webinar platform, we do want to offer you more ways to connect with each other and solve problems together. After all, that's part of why we're all here. So to help connect you, we created a Slack group channel for the global network. If you didn't receive our newsletter earlier this week with the link to join, please contact hello@biomimicry.org and we'll invite you to that group to get started. The Slack group serves as a safe place to exchange ideas and resources, so please be mindful and respectful to your fellow nature lovers and innovators on this platform. If you're tuning in with us on YouTube Live, you can participate in the conversation on Twitter and Instagram and use the hashtag #BiomimicryChat. All right, so I'm going to close the poll here. Wow, so it looks like, let's see, I'm going to share it with the group here. It looks like we have a wide range of challenges to solving design problems. This looks like a great place for us to begin this journey so we can hopefully work through solutions to overcoming these challenges together. Today, we're going to dive into the process of medical problem solving and see how you can find inspiration from nature in many different ways. From the biomimicry design spiral to free-form bioinspiration, where many have stumbled upon some novel mechanisms that can guide us towards sustainable solutions across industries. Bringing us into these two realms of thinking today is Jeffrey Karp, a professor of medicine at Brigham and Women's Hospital Harvard Medical School. Jeff is a master problem solver and holds over 100 patents, has launched 10 companies with over $400 million in funding that have generated several products currently under development and on the market. And Alexandra Ralewski is the science director at the Biomimicry Institute. She brings her love for biology and passion for design thinking to educate the public on the creative potential nature offers. Before joining us at the Institute, she facilitated translational research in plant biology and neuroscience at Yale University. Jeff and Alex, welcome, and thank you for being with us today.
Awesome, thank you so much, Lex. Yes, thank you for those wonderful introductions. And Jeff, again, thank you so much for being here and taking the time. It's wonderful to see you again and to have this opportunity to talk about what are two of my favorite topics. I'm assuming there may be two of yours as well, nature and innovation. I just believe there's such an enormous opportunity for nature to promote and grow and feed innovation by providing us with solutions that we really never could have imagined otherwise. And so talks like this, or fireside chats, if you want to call them, it's middle of the day for me, sort of afternoon for you. I think these kinds of talks are a really great opportunity to dive in and explore these topics and really give people an understanding of how looking to nature works. What's that process that's involved? What's that pipeline that goes from research all the way to market? But before we dive too deep in, I wanted to start off on sort of a broader level, because Jeff, as Lex mentioned, you really work to create advanced biomaterials and devices for therapeutics to really improve patient care and to really help people. But in your lab, it's special in that you look to nature to help you solve some of these problems. And so I thought I'd start off by asking you about what was that first time that you looked to nature? You know, what was that light bulb moment that sort of went off and you were like, aha, you know, there's this whole other library of solutions and knowledge out there that I could tap into. So yeah, please tell us a little bit more about that.
Absolutely. So it was back when I was doing my postdoc at MIT. This was in probably 2000, late maybe 2004, 2005 timeframe. And I was walking around the lab one night and just kind of looking around, and I saw Spider-Man in a journal on someone's desk. It was just open, and there was this picture of Spider-Man kind of hanging, an action figure actually hanging from the ceiling.
Right. Some light reading for the late night researcher. Yeah.
And I knew it was a nature journal, and I was like, you know, what's going on? So I went back to my desk and I sort of, you know, looked up Spider-Man in nature, and I found the article and I read through it, and the authors had basically, through understanding how geckos can attach to surfaces, you know, just in their surfaces of their toes, this group had mimicked that and created a tape, which they then applied to Spider-Man. And so really the key kind of action figure, if you know, so to speak, in the paper was Spider-Man stuck to the ceiling. And that was, to me, that was just really fascinating and shocking, and, you know, all kinds of different things together. At the time, it even struck me more because at the time I had just been kind of going around to a bunch of doctors and talking about unmet needs, and realized that there were huge unmet needs in developing better surgical adhesive. So not necessarily materials that could attach to the skin per se, but actually inside the body. So materials that could seal the lung, for example, or seal the GI tissue or the heart. And it kind of occurred to me that the requirements of a adhesive tape that would be used inside the body would be very different than something you'd use, you know, to attach Spider-Man, let's say, to the ceiling. Right. And yeah, and so that's how it all began. And you know, I was so fired up about it, and at the time was kind of developing some materials with a team of people. And I discussed, you know, this concept with them, and there happened to be a request for proposals at MIT to go to the National Science Foundation, and we cobbled a proposal together to create a biodegradable, biocompatible, you know, adhesive tape that could be used internally in the body. And we got approval through MIT, and then eventually got the grant, and that's how I got into this space.
Wow, that's amazing. And so, yeah, I'd love for you to talk more about the different people in your lab, because I know they come from such a wide variety of backgrounds. I know you said you have bioengineers, you have chemists, you have medical students. And when you're trying to solve a problem in nature, you know, all of these disciplines, they have their own method of problem solving, right? The way the engineer solves the problem is different from the way the medical student solves the problem. And none of them are really looking to nature. So how do you go about that process of having all these different disciplines come together and look to nature to solve that problem? And how has, when you first started, you know, you first stumbled upon, you know, this Spider-Man in nature, but how has your process sort of evolved to look to nature specifically now? Yeah, and how has it improved?
When I started my lab in July of 2007, I really kind of set off with two main goals. And this was based on, you know, just having really great experiences in my PhD and in my postdoc. And so mentorship was one of them, professional development of people in the lab. And so I really wanted to make sure I was able to create a conducive environment where people really were comfortable and empowered and free flowing ideas, and, you know, people could make mistakes and learn from those mistakes, and it just sort of, you know, that was kind of part of the process. And the other main goal was translation. So I had seen my PhD advisors as well as my postdoc advisor start companies and move technologies from the laboratory you know, into clinical trials, and then eventually to patients. And I just saw that as really important, and something that was really, you know, a powerful thing that we could do as academics, was not just, you know, publish papers in journals, but rather, you know, set things up so that we could maximize the potential to help patients in the shortest period of time. So I made that really the second major goal of my lab. And sort of as I was going, I sort of realized that there were a lot of decisions I had to make and could make. And one of the things that just really hit me was this opportunity to not necessarily follow in the footsteps of others that I had seen before, or, you know, and sort of follow a direct path per se, but to really try to make it authentic and really sort of align with my passions and what I thought was right at the time, which was, you know, I really wanted to create a multidisciplinary lab where there was minimal overlap in the expertise of people who were there. So if you can imagine, like, you know, if we are advancing on a project and we encounter a challenge, you have a bunch of people sitting around a table and everybody can bring a unique perspective because there's minimal overlap in the expertise. Right. And then everyone immediately feels validated because they've brought something, you know, different to the table that really no one else can bring. And then I feel like when people feel validated, they're more likely to go all in and just really, you know, kind of give it their all. And so that's where I really see kind of this multidisciplinary, you know, aspect of the lab being very, very powerful. It also, you know, kind of does create some friction because, you know, we will have, like, just as an example, I mean, we have engineers and biologists. We've had a cardiac surgeon in the lab, a gastrointestinal surgeon. We've had a dentist, multiple MDs, and, you know, not everyone is sort of comfortable talking with one another kind of fluently, at least in the beginning. But, you know, I have this sense that when you create an environment that sort of makes it essential to communicate, and also one where there's very few limitations on people. So people can contribute to any project they want in the lab. You know, usually people have one or two core projects, but they're open to interact with anyone else. And there's really no competitive projects at all in the lab. You know, every project is unique. And so one week, you know, we might be talking about tissue adhesives, for example, the next week, cancer, the next week, arthritis, the next week, you know, some surgical device that we're developing. So, you know, it's very, very kind of different every week. And the other thing I'll just add to this, which I think is really important from a problem-solving perspective, is a multicultural environment as well. So we've had people from over 30 countries in the lab, and I feel like, you know, everyone has from different countries, different education systems, you know, different problems that they've seen, you know, growing up and that exist in their country. And so, you know, that kind of mixture of different perspectives, I think, you know, what we're doing is really difficult. You know, it's one thing to publish a paper, it's another to, you know, have a technology form a product that then actually works, you know, on patients. And there are a lot of challenges that we have to address along the way. And then finally, the kind of tying in bioinspiration, the way that in biomimicry, the way that we have, you know, tried to incorporate that into the process is not necessarily starting with it as a main goal, but rather really focusing on defining the problem we want to solve, and figuring out what's the first experiments we can conduct to gain some insights. And then as we advance, when we hit a wall, which we always do, you know, encounter a major challenge, then we turn to nature for inspiration. And that's where nature has really been able to help us the most.
Yeah, it's great. And, you know, I think having the baseline of that translational research and sort of everyone starting at that point, I think is so key, because it's something that, you know, we talk about in research and academia all the time, oh, translation, but it's really between, you know, you and the lab next door. And so when I was doing research, you know, I came from a plant biology background and I was in a neuroscience lab, and they were all neuroscientists. You know, maybe they came from, you know, slightly different labs and they focus on, you know, different areas of the brain, but really that was their one specialization. So for me to come in with a totally different background and provide like a totally unique perspective was really eye-opening for everyone. And what we're talking about here is taking it even a step further to go outside the lab. You know, everything that we do is really about, you know, model organisms and really how can we use nature a lot of the time. And here it's sort of saying, what can we learn from nature? What can we adapt? What can we emulate? And I think having people, you know, regardless of what background you come from, you can have nature sort of be that grounding point and that sort of starting point that everyone can learn from. So one of the examples actually that I'd love for you to talk about a bit more and sort of diving a bit deeper into that process is the porcupine quill staple, which is one of my favorite inventions of yours. And I just love if you could talk a little bit about, the walls you came up against, how you, look to nature, what your different sources of inspiration were, and then, how that product eventually became successful for use.
Yeah. So initially we were working on the Gecko inspired biomedical adhesive, and we advanced that to a point where we got it up and working in some preclinical models. And then we were aware that not a single tissue adhesive was going to solve all the problems, because there's many. And so I was brainstorming one day with a colleague, a friend of mine, Rohit Karnik, a professor at MIT, and we were just started talking about, I don't know how we got on the topic, but we were just thinking about nature. I think probably because of the Gecko inspiration of the first project.
Right.
And the conversation was just about, how do various creatures latch onto their hosts, and how does, can we think of maybe something that has been unexplored that would make it even more exciting, to advance. And we were looking at like the proboscis of mosquitoes and,
Right.
bees. And I think it was Rohit who said, well, what about porcupine quills? And so, I kind of went on my computer and started looking things up, and quickly realized there's, a few different types of porcupines. The North American one kind of jumped out at me, because the quills on the porcupine have these backwards facing barbs, so they face away from the tip. And so what happens is when the quill goes into your tissue and you try to pull it out, the barbs kind of bend backwards and sort of drag along the tissue fibers and really get stuck, which makes it really difficult
so cool.
to remove it.
As many dogs know, yeah.
Oh yeah. So if you Google, if you just Google porcupine quill on Google Images, you'll see a ton of dogs that have, had a unforgiving encounter with a porcupine. So we went to the academic literature and we started looking it up, and we were amazed to find there were very few papers that described the mechanism of how porcupine quills can get into tissue. And we kind of knew that porcupines, they don't run at their kind of target. They don't try to engage like that. They pretty much just kind of stand still. They're slow moving creatures. They can kind of whip their tail around a lot, but that doesn't really release quills. It's quills are not really flying through the air type of thing.
Right.
So we knew there had to be some mechanism for them to insert really easily into tissue. So because we didn't see too much in the academic literature, we were immediately excited to say, okay, well, maybe we could put a project around this and just start asking some basic questions about how they work. And so what we did is we bought some porcupine quills from eBay. A lot of people use them there for artwork.
Okay.
So we bought them from there. Actually, at one point, my institution, because in our paper we said that we obtained these porcupine quills, and they thought that we were working with porcupines, and we hadn't gotten like the animal approval. Yeah, running around in the labs. You were like, no,
Aya Cook was all over you. Yeah.
They were kind of they're the quills on their backs were raised. But so basically what we found was that we took these quills, and the tips of the quills, usually it's about very small, 4 or 5 millimeter section that has the barbs. And we started like sanding off barbs in various regions, and the whole thing. We did comparisons, with and without the barbs. And what we quickly realized is that the barbs endow the quills with some incredible properties that we really hadn't appreciated before. So we already knew that barbs can really latch onto tissue and make it hard to pull out. But what we discovered as well was that the barbs actually reduced the penetration force into the tissue. And so it required about half the force to push a barbed quill into tissue versus a needle of the same diameter, or a quill that we have kind of gently shaved the barbs off. Right. And we did some sort of modeling to figure it out. And what we think is happening is that when you go to cut a tomato, just as a parallel, you use a serrated knife so that you're cutting really just the surface of the tomato. On the other extreme, if you have a dull knife, you're kind of crushing the tomato as it's going through. And what we think is happening is the barbs are kind of just, kind of in a radial fashion, are cutting the tissue fibers as it goes into the tissue. These little barbs are raised off of the quill kind of surface. And we think it's like cutting the tissue. And then, so not only do we find this reduced force, which we were amazed by, was really incredible, half the force. But when we put the quills into tissue and then we cut it, it had a perfect, like the tissue was right up against the quill. There were no tears. It was like a perfect hole that it had created. And if you take a quill without barbs, and so you shave them off and you put them into tissue, it just tears the tissue on the way in. Same with staples or any needle. If you put it into tissue, it actually tears the tissue on the way in. So it's not a perfect hole. And if you're talking about staples, then bacteria can get in between the staple and the tissue. And so that's why you typically have infections on staple tracks, you know? So we thought, okay, well, what if we took kind of standard staples, which you push into tissue and then you have to bend it to hold into place. What if we made those staples have quills on either end? You push it into the tissue, so you immediately it grips the tissue. No need to bend the staples,
right, because it just stays in, yeah.
It just stays in, yeah. And then if you make it biodegradable, you never have to take it out. And so, so we've been working on trying to advance that. And one other thing I'll add to this is that one of the big challenges actually here that we've faced, and this is starting to kind of come online, but it's how do you manufacture synthetic quills large scale? These barbs are on the order of like, the size of a human hair, 1 or 2 human hairs. And so there really hasn't been, even with 3D printing, ability to create, millions of these in a cost-effective manner. And so this is actually slowly kind of catching up. And we hope that these kind of new manufacturing routes that are now kind of coming online will allow us to take some of these discoveries and then bring them forward.
Oh, excellent. Yeah. So you always have to think about that pipeline down the line as you're starting that initial development. And so one thing I wanted to ask you is that, why did you choose the porcupine specifically? You were looking at parasites, you were looking at mosquitoes. There are plenty of things that go in to skin and to tissue and to wood. Were you looking for the additional quality of that it couldn't come back out? Or what sort of led you to sort of say, what, this is what we're choosing, let's go down and see what we get?
Yeah, so I think part of it was so I would say majority of the projects, not all, but majority of the projects in the lab really start from a problem, and then conducting experiments to better understand the problem and gain some insights. And actually sometimes that can come from, you have a problem, you come up with a half-baked solution, you try it, but the goal is to learn, not necessarily to succeed, because you rarely succeed the first time you try something. And so, then the initial parts of the project really focus on understanding the problem. And I find that, usually insights emerge, and then those insights kind of point you towards solutions. And so that's how we conduct most of our projects. But sometimes, we just let our curiosities run wild. And it can be a lot of fun just starting with a creature in nature and just trying to mimic it. We had in kind of a parallel project, what we did is we went to Google Images and typed in parasite, because parasites have all kinds of nifty ways of latching onto their hosts. In fact, they require that for survival. And we found this spiny-headed worm which inserts a needle-like proboscis into the intestine of fish. And then the top part kind of swells and then it locks into place. And as soon as we saw that, we're like, well, I wonder if we could make something synthetic that can do the same thing. So we actually figured out a way to do it. And we created this Microneedle Patch that actually now has led to all kinds of different projects in the lab. So this patch is, instead of, the worm uses these muscular structures to swell the tip after it goes into the tissue. What we did is we took kind of diaper-like material that is super absorbent and can swell. So we made the tips of these needles have that polyacrylic acid. When you put it into tissue, water is ubiquitous in our body. So the water goes into the tips. It starts off really hard and sharp, so it can go into the tissue, and then it starts to swell, and we got it to lock into place. And so the porcupine was really just, kind of a parallel project where at the time we were just super curious about different creatures like attaching to tissues. And we knew from all the surgeons and other doctors we spoke to that there were a ton of unmet needs to develop better tissue adhesives in medicine. And in fact, at the time, really the best that we had was medical crazy glue, It's a material called cyanoacrylate, and it can attach very strongly. If anyone's kind of gotten crazy glue on your fingers, you're in trouble, right? Because it just sticks so strongly. The problem is also highly inflammatory. And so you can use it outside the body on the skin, and you can go down to like CVS or Walgreens or wherever and get some. But if you put it inside the body, it just causes a massive inflammatory response. And it also, you have to keep the tissue fairly dry when you put it on. So that kind of speaks to the significant limitations. And sutures, you have to realign the tissue each time you pass the suture needle, and that can take a really long time if you have a big incision you need to close. The longer someone's on an operating table, the more risk of complications. And Staples had issues. So we just knew there were so many unmet needs for new adhesives. We kind of started to go a bit wild and just like, kind of look at nature and
what's out there.
What's out there. Yeah. And just let your kind of curiosity flow. Yeah.
And that's the beautiful thing about about biomimicry is that, people say, okay, how do I do biomimicry? And it is a practice that, people spend their whole lives, like myself, I practice biomimicry, but it's also a process, it's a problem solving process. I think it can be so much fun to say, okay, how does nature attach? And then, one of the tools that we provide for people is Ask Nature. And it's just an online library that has thousands of these organisms, and it's organized by function. So you can go online, you can say, okay, how does nature attach? How does nature adhere? How does nature, protect from temperature? And you can go and you can click on that, and you can get you get the porcupine, you get the parasite, you get all this. You spend hours and hours and hours just searching all of these beautiful organisms and just being, amazed. Really, I work on Ask Nature a lot, and I'm still discovering new organisms and new different ways that that they have just beautifully adapted to their environments. And so having tools like that, I think, is incredibly important. Lex also mentioned the design spiral, which is another tool that sort of helps people walk through the process of biomimicry, of starting with a design and then saying, what can we emulate to apply, to medical staples, to medical needles, that sort of thing. So,
I would actually just say, kind of reflecting on what you just said, and kind of building on it, I'd say, we need biomimicry more than ever. We need to be closer to nature, I think, than we've ever been. And I think, there's many reasons for that. I think, some of the reasons that, I think about quite a bit are, we've kind of created our, society, civilization based on existing within these four-walled structures, like concrete jungles. And I think that this really reduces our ability to solve problems. It really negatively impacts our ability to explore our curiosities and be creative. And I think over time, we kind of just become a product of the environment that we're in, where I really feel like all of us were meant to exist within nature and be very close to nature. And I feel, it's one of these things, whenever you go on like a hike or you get a chance to get into nature, it's just like, like so refreshing, and it's so just feels right, And
are you looking now every time you go into nature, you say, oh, what's that? What's that bug doing? What's that bird? Let me go home and Google it or go on Ask Nature. Is it sort of, now that you've taken this approach, has it sort of changed the way you see nature and then think about organisms?
It does. I think, I don't know, I'm kind of the type of person where my curiosity just goes like berserk, and then I kind of get like stressed out because I want to know too many things, and so I have to kind of tame
like 100 tabs open on your computer.
Yeah. It's like when I walk into a bookstore, I try not to too often, because there's so many things I want to read, and I just can't really make the time to read all the things I want to read. So it kind of stresses me out. I feel the same thing in nature when it's kind of, so I try not to try to just be at peace when I'm in nature and just be sort of relaxed and not really put on my sort of work. I kind of see it as an environment that can complement the work I do, but I try not to think too much about work when I'm out in nature. Just try to more embrace it.
Yeah. Just sort of let it soak in, and then, when you go home, you say, oh, maybe I remembered that thing. I remember that woodpecker, and
exactly.
So I do the same thing. I was recently camping, and we were on this beach, and there were these moon snails, which I had never seen before. And the little hut that they used to encase their larvae, if you pick it up, I actually thought it was garbage that someone had thrown on the beach because it just looks like rubber, like human rubber, and it feels like rubber. But it's actually made by this organism. It's absolutely astounding. So I have to go on to Ask Nature, and then I'm actually going to also Google and see if I can learn more about the moon snail, because it's just, it never ceases to amaze me, the things that nature creates. So one of the other questions that I have for you, and that has come up a lot, is if you could really help describe for people that process of, say, okay, you have this great idea, you're going to do the porcupine quill, you're going to make this, this medical needle. How do you get that to market? Like Lex mentioned, you have so many patents, you have so many companies, you're clearly very successful at this. I'd love to get your take on, some of the pitfalls that you encountered. What are the relationships you need to establish with other people to get that going? And then also speaking to your idea of radical simplicity. I know that you gave a TED talk about it. Lex can probably drop that in the chat, or can email that to people after. But you have these 3 principles for radical simplicity and how you can start to approach these problems. And I think it's a really wonderful framework for thinking of not just for biomimicry, but any sort of problem solving. So yeah, if you could speak to that pipeline and how radical simplicity relates to that, I think that'd be wonderful.
Sure. So when I started my faculty position, as I was kind of mentioning a bit before, I really wanted to figure out a way to maximize the potential for anything we worked on in the lab to actually help patients in the shortest period of time. And I knew from the mentors that I had, in my life, my PhD mentors and postdoc mentors, that that there actually was a way to take academic discoveries and bring them forward. But it's one thing to kind of watch other people do it, and it's another thing to kind of do it yourself. And I quickly realized that I lacked, as an academic, core business training. I just really didn't know, and there were just I really yeah, you're not taught that, and I feel like I spent a lot of time in self-awareness and trying to figure out, like, what is it that I really know and what is it that I don't know, and what are my strengths and weaknesses, and who around me has strengths where I have weaknesses, and things like that. And so I quickly realized that the only way that I was going to be able to kind of fulfill this mission of translation was to develop relationships with people in the community who were really good at it. And as I kind of advanced, I realized that there were so many different areas of expertise that are essential to bringing these basic discoveries, along this translational path and turning them into products. So what I did is I literally, from the last 13, 14 years, and maybe even a little bit earlier than that, I have made a commitment to meet someone new in the entrepreneurial ecosystem every 2 or 3 weeks. Be it like a patent. Yeah, it's a big commitment, especially when writing grants and, all these other things kind of going on. But I just, even the first few years, I decided I wasn't going to attend that many conferences. I just really thought this was important, and I made a lot of time for it. And so, I would go out and meet like patent lawyers and corporate lawyers and people who knew about manufacturing.
Call them up or email them or get references from other people, or?
Everything. I mean, I would go to talks where I knew someone was there who I thought, might be a good person to develop a relationship with, who could help. And then, I would try and ask a question and then go up afterwards, and, and then I would just like cold call people or cold email people or ask for introductions. And just really, I realized that a lot of the law firms in town had networking events that, because they wanted, new companies to use their law firm. So I just got myself on lots of invite lists for all these law firms, and just anything where I could kind of intermix with people and just, kind of chit chat, at various events and things. And what happened is that it wasn't, I didn't look at it as kind of like a one-time meeting. I looked at it as, I'm not going to be able to maintain kind of relationships with everybody, but there's probably going to be a critical mass of people that I will be able to if I really make a big effort. And so that evolved. And then what started happening is those people that I started developing relationships with, ended up serving in some ways almost as like this advisory board for my lab, because like I would start asking them questions like, you know, we're going to be making this material, and do you think we could make this on the large scale? And, you know, they would say, you know, don't, you know, if you're going to require a 5-step synthesis, like that's going to kill it because you have to do quality control at each step. Can you get it down to 1 step or 2 steps?
It's such valuable feedback to have from the get-go, you know?
Yes,
Wow.
yes, exactly. So it was that kind of thing. And that's how, and then the other key thing about that, which was really critical, was, you know, I noticed a lot of my colleagues would sort of advance and make a discovery, and then they would look to translate, maybe form a company, and you need a CEO of your company, an entrepreneur. And I realized that, you know, it's very difficult to form a relationship quickly with somebody. And so, you know, and unless you know someone really well, it's going to be tough to really make it work out, because it's just so difficult to do, you know, bring products forward in new companies. So through these interactions, I made a habit of kind of meeting new entrepreneurs and CEOs of companies. And so as ideas were advancing, I would be talking kind of in this network to various people. And then as an idea got more and more mature, I was able to say, hey, are you interested in being involved? Do you know someone who might be? And so that ended up, you know, it's almost like having lots of potential CEO candidates that were now kind of in my circle who could then help to drive it forward. And that's actually really been the model that I've advanced, I think, the most, is partnering with an entrepreneur, someone who has deep business expertise, who can bring, you know, really creative and great people to the table, who are experts in, you know, reimbursement and regulatory and manufacturing and patents and all these things, and then they can assemble them around the table, and then they can go out and help to raise money. And then that really, you know, helps to create the momentum. So I'm always looking for ways to create more and more momentum.
Wow. Yeah. And so do you find that when you know, how do you get other people, like on the other side, you know, other physicians or other clinicians involved in this, where they say, oh, you know, what's this nature inspired solution? Or, you know, I'd rather go with a traditional engineering solution. You know, what's the process for getting them more involved? And what do you think the value of exposing them to this type of research and design thinking is?
I think that clinicians and kind of bringing people in, that they're very interested kind of at first in solving the problem. And you know, one of the things that we focus on a lot in my lab, and something that I ask almost every single lab meeting, is what's the bar that we need to exceed to get people excited? So in other words, you know, what's the best result that anyone has ever achieved with a particular technology or specific kind of disease model? And then how much better do we need to do to really, you know, get the world excited? Because you need that kind of momentum and that excitement to bring things forward. Because, you know, these technologies require often tens or hundreds of millions of dollars to be able to kind of, you know, bring it to patients. And so by having that initial focus and really making that the target of every project, when let's say we go to talk to collaborators, you know, other clinicians and things, to kind of get them involved, you know, often, not always, but often they'll be most excited about, let's say, the data we have in hand, or the potential of the technology that we're developing to do better than what currently exists. And when we have a bio-inspired solution, and we don't for every project, but we've turned to nature for many of them, it kind of adds that additional sparkle, you know, to the project, that just makes it more interesting and kind of pulls people in. People are more curious, and I think that you know, curiosity really is free flowing when you're in nature, and I think when you start to incorporate like a nature, you know, bio-inspiration or biomimicry into projects, I think, you know, people, it kind of draws people in. People love stories. They love, you know, whether they admit it or not. I just feel like everyone has this relationship with nature, and so when you start talking about it, it just has this ability to draw people in.
Absolutely. And it works, you know, that's a beautiful thing. It's, you know, it's already a proof of concept in and of itself. It's been tried, it's been tested, it's been validated. So to sort of say, you know, we're using this thing that clearly has already been successful in this kind of environment under these conditions, you know, you're already starting with something that, as you know, is at a technology readiness level of 3 or 4, which is not something you can really say for any other solution that's, that someone just thought of off the top of their head. And so I like what you said about sort of establishing that territory and saying, what else have other people done? Because I know that's step one of radical simplicity. I was hoping you could discuss the other two steps with people and just sort of give a bit more context for that.
Yeah. So, well, I think the way that I really think about radical simplicity is, and this is actually, I learned this kind of the hard way, which is when I started off my lab, I talk about this in the talk that you mentioned, this TEDx talk that I did, but came up with a solution and got some really compelling data. At least I thought it was compelling, and went to a venture capitalist in town and said, "Hey, let's start a company." And they basically looked at me and said, "This is way too complicated. It's never going to help anybody." And I had spent like 2 years of my, it was like the first project in my professorship that I worked on. So I was kind of devastated. But that was like one of the most important lessons that I have ever learned, I'd say, in my professorship. But I think, I really think of, and that's where this concept of radical simplicity was really born. It's really about minimizing the complexity at every step. So complexity only when it's necessary. Some solutions need to be very complicated just because, there's just, at least with present thinking and tools we have available, it's tough to simplify, but it's really kind of simplifying at every possible step you can imagine. And manufacturing kills so many potential technologies because, you can get something to work, let's say in a preclinical model, but then when you start to advance it, you realize it just can't be manufactured or it's too costly and I think that I'm still trying to come up with ways of how we might use nature to improve on manufacturing approaches. And I think, there's probably some pretty interesting things out there and just starting to kind of look into that.
Yeah.
But I do feel like maybe one thing that we kind of face, as we're trying to translate that nature hasn't faced, or maybe, I just need to look at it in a different way, is this sort of cost-effective scalability. We make products, unfortunately, often, in the world that are non-degradable, and they don't really, they shouldn't be within the ecosystem. They don't contribute to the ecosystem. Whereas if you go into like the rainforest, for example, it's like everything is relying on everything else. And
it's a fully closed system. Yeah.
Yeah. So I think, so I think, there's a lot to be said, and that's one of the challenges we often face that I think we really need to think a little bit more about, and perhaps nature has some secrets that can help us, which is, how do we take solutions and manufacture them, as simply as possible, and to make them, cost effective. It's been a big challenge and has killed a lot of things. So that's why we think about that early on and use it to make decisions in our projects.
Yeah, there's definitely solutions that can be learned from nature with regards to scalability and manufacturing. Materials are a big thing that come up a lot, and I think is sort of a hurdle that people come up against. Like you mentioned, they have this great idea, it's bio-inspired product, but then they use, plastic or something to develop it. And sometimes that's the only material that's available, but there's so many materials in nature that just haven't been explored for all of their different properties, and figuring out how we can use those or how we can synthesize those more effectively to get, much more efficient and, reusable materials, I think would be fantastic.
Actually, something to add to that is in March, I was asked to co-lead this N95 respirator work group for Mass General Brigham, and essentially, it was to create masks for clinicians, of frontline healthcare workers. And if you look at, you can imagine how many of these exist, how many are being produced right now. And these are non-degradable kind of permanent materials that are going to be sitting in landfills for hundreds or maybe thousands of years. I don't know how long it takes to degrade, polypropylene type materials, but it's, a major challenge for us to think about. And there's materials that we can make masks out of that are more environmentally friendly. But I think it's, the world really is focused on sort of the cheapest solution possible, regardless of its impact on the environment, unfortunately. And so it's really hard, even if you have materials that could be a replacement, it's hard to get people to pay for that, and to draw, just with all these budgetary constraints and things. So, I think, we need a lot more. There's a lot more we can do, in this. I have some ideas, but it's something that bothers me a lot. I'm really thinking deeply about the environment and how we might be able to start some projects up soon, in the lab on this.
Oh yeah, I mean, that was actually one of the questions we had from the audience. They sort of want to know, like, what you're working on now. Are you working on anything, COVID related, as you mentioned with the masks? But what has you excited now? What are you looking forward to in the future for your research and your work?
We're doing a lot of COVID related work. So masks is a big thing, and I think it's also important to consider that, there's other, while things are starting to get a little better in the US, specifically because most people wear masks, and I think we'll kind of hit like new steady state, but then as people kind of feel more freedom to do whatever they want, we'll see like kind of waves, hit. But if you look at like Brazil, for example, they're just getting hammered right now. And there's other regions of the world where we just need to get new resources to as quickly as possible. So we've also been working on disinfection technologies. We've been working on repurposing drugs to try and reduce the severity of COVID. COVID tends to really it hits certain people, and we don't know why, and makes them extraordinarily sick and require ventilators. And there's something called a cytokine storm. So cells essentially just, neutrophils can just release their contents, and they have all these cytokines, and it's just, you can't, the inflammation process is run so, it's just like an explosion that you can't control it. And so we're working on kind of repurposing of a drug to try to prevent the intensity of these sort of inflammatory responses. So, that's something that we're working on, and we're developing a gel that can go into the nose. And it's one of the entry points of the virus is in the goblet cells in the nose. And so we have a spray that we've created that has antiviral agents in it that could, capture the virus and then kill it. And, it's not none of these solutions is something that would be like a standalone. It would be used, in combination with other protective means. But we're doing, and even a detection system, some part of a team that's come up with a completely new way to detect COVID. And so we're in the process of, kind of putting that together and moving ahead with proof of concept experiments. But we're doing all kinds of different things in the lab. I mean, one technology is we have a needle that automatically stops in between the layers of the eye, to deliver gene therapy to the back of the eye, to treat all kinds of different diseases. There's been remarkable advancements in gene therapy, and now it's a viable therapeutic approach in the clinic. And but it's really difficult to get drugs to the back of the eye. So we came up with a needle that can help with that. We're doing a lot of work, a lot of different things. We have a grant from the NFL Players Association to help with osteoarthritis. So injury in the joints can lead to osteoarthritis. And we have a gel that we developed with a certain therapeutic that can slow down the progression of the osteoarthritis. And so we're working on that, and we have a bunch of projects in inflammatory bowel disease and all kinds of things, even diabetes, type 1 diabetes, type 2 diabetes. We have a pill that you swallow that coats your, you take it before a meal, and it transiently coats part of your GI tract to reduce nutrient exposure, trying to mimic the beneficial effects of gastric bypass procedures on type 2 diabetics, which has really been remarkable. But it's very invasive surgery that you need to get these results. And so we've tried to create kind of a surgery in a pill type approach.
Wow.
So yeah, doing all kinds of
It's a huge
things.
lab. It's a lot going on. So now with COVID, how do you do that research? Are you just spending a lot more time sort of online looking for, different inspiration and try to advance it in that way? Are you doing a lot more of that upfront online research now while you're not able to be in the lab?
Yeah, the lab is, kind of came to pretty much a standstill for the last, several months, and in early to mid March, we had to close it down, and we've just been, the institutions in Boston are kind of just slowly getting back to normal. We've had to separate the lab into two groups. So one comes in, Monday, Wednesday, Friday. One comes in on Tuesdays and Thursdays, and so that's how we're starting to move some things forward. But it's actually been there's been quite a few challenges actually that we've been hit by. One is we had a hiring freeze, so we couldn't. A bunch of people were supposed to come for the summer, and you can imagine they booked their plane tickets and they were ready to go and really excited so they couldn't come for the summer. And now there's a lot up in the air right now about how we can kind of recruit new people in and to the lab, but hopefully that'll get going shortly. And some companies that were going to fund the lab ran into financial trouble, and so they pulled out there. So we're kind of experiencing challenges on different levels, but we're still trying to keep our heads high and move as keep
and it's a good opportunity to sort of do that online work. It's great that you mentioned cytokine storms, because we actually just created a new collection on how does nature protect from pathogens. And we're sort of taking that other approach of saying, okay, well, why do we get infected with viruses versus bats, obviously, who are known to carry a lot of these things, and sort of understanding how they basically, their immune system dampens the response, and basically they don't experience a cytokine storm. And so,
by the way, just on that one point, just to kind of add to that, I'd say, a lot of people are talking about vaccines and how they, if they work and they're safe, they really could make a huge difference. But what I think maybe a little bit underappreciated is drugs that can tone down cytokine storms, that can tone down this massive inflammatory response. If you can really do that, you might be able to turn COVID into kind of like a common cold. And so you wouldn't have these severe, effects. And the key is, though, you have to catch it early, and you have to have a drug that's very safe, because you really don't know early. I've talked to a lot of doctors who treat COVID patients, and they don't know which ones are going to need ventilators when they come in and which ones aren't. Like it's impossible to predict it right now. So exactly. But sort of saying, okay, well, how are these other organisms solving this problem? What proteins are being activated, what pathways are going on that are sort of preventing some of this, and saying, okay, what's similar in our bodies? What can we mimic? What drugs or vaccines can we develop? And so that's why we sort of created that collection on Ask Nature, which is going to be launching actually next. There's going to be a new sort of soft launch next week, where we have these 3 new collections where we're talking about pathogens, how does nature encourage resilience, and how does nature create community? So all of these things that people are asking about and looking for right now, we wanted to address that, especially at this time. So people can tune in for that soft launch next week. Cool.
And then just a little bit of more of more self-promotion for Ask Nature, just before we wrap up here. We're also going to be doing a massive relaunch, total overhaul in September, which is going to be really exciting. Going to have hundreds of new strategies, a lot more innovations featuring, research that's done by Jeff and a lot of other companies and industries that are now looking towards bio-inspired design. So we hope that people will look out for that, in the coming months. And so, Jeff, one more question that I wanted to ask you is, what are some other areas, I know you don't like predicting the future, but what are some other areas that you really think could be helped by biomimicry? I know you focus a lot on, medical research and therapeutics and patient care, but what are some other areas that you see, people do a lot with robotics, they do a lot with optics and, structural color for, reducing harmful chemicals and paint. What are some of those stories that you see that excite you for the future of biomimicry?
Wow. I don't know. I just, I tend to think of, the sense, I think we were talking a little bit before about, predicting the future. And I think the only thing I can say is that you know, humans are going to create more problems than they solve, right? And I think we'll continue to do that. And even more reason that we're going to need to turn to nature for inspiration and really get into the habit of making that you know, a go-to process. You know, just as a way to maintain, you know, our curiosities and to really, you know, maximize creativity. And you know, I don't necessarily have kind of specific examples, but I in my experience, it seems as though every solution we've come up with, you know, if we haven't used biomimicry, at some point we see something in nature where it had already been solved. So it's almost like, you know, you know, there's so much in nature to learn from, and I think we're really at the tip of the iceberg, because what it really takes is, you know, there's so many solutions out there. And we just need to do, you know, we need more research on those solutions so we can better understand the mechanisms that exist around us and sort of expand that library of potential solutions that we could then draw on for all the different problems that we're going to be faced with, you know, not just today, but into the future. So I think it goes beyond just looking at nature. We actually need more people exploring nature and, you know, kind of discovering new mechanisms, because all of that will come back to helping solve, you know, human and animal and, you know, environmental problems that we're all faced with.
Absolutely. And that's why it's so important to do that research. You know, I know a lot of species are disappearing and are becoming endangered, and it's so important to make sure not only to save them and to save their habitats, but also to make sure that we understand how they've adapted and what we can learn from them. That there's just so much value there. I think it's also additionally important, you know, when we think of innovations, that we're also really focused on the fact that, you know, these problems, when people ask me, oh, you know, Alex, there's this problem I haven't solved, there's this problem, you know, and I'm like, there's never been a problem that someone's presented me with that I haven't found a solution in nature to. You know, maybe it's not maybe always a one-to-one, you know, maybe it's a material thing, but there's always some solution in nature that they've already come up with it first. So we don't need to reinvent the wheel, you know, and I think it's always important for people to keep that in mind. And, you know, that I was actually doing, I was on YouTube for some reason. Actually, there was this ad that came up, and it was actually the first YouTube ad I've ever watched. And it was from Microsoft, and they were talking about biological computation. And basically a big problem that they're facing is the fact that they need to store massive amounts of data, and they also need to transmit massive amounts of data. And that uses so much electricity, uses so much carbon emissions. I mean, it's starting to become a, you know, rather large problem. And they realize, well, you know, what's the best at storing large amounts of data and transmitting that? Well, it's DNA, and it's, you know, living organisms and cells. So they now have created this whole field called biological computation that really looks to understand how living organisms and cells store data and transmit that information. And that you know, that small piece of DNA with those 4 letters has the capacity to transform an entire field and, you know, directly impact the way that we experience data and store our data and, you know, share information with everyone around the world. So a lot of times when we talk about biomimicry, it's this, you know, one-to-one sort of, you know, and it's very valuable. So, you know, the porcupine quill and the staple, and that has massive power to transform lives. But I always want to make sure that people understand that there's many levels of biomimicry. You can go down from the smallest cell and DNA all the way up to, you know, an entire ecosystem. And every level in between is something that we can learn from and emulate and really use to transform massive disciplines. So I see that Lex is back on here, so I'm gonna hand it over to her. But first, I just wanted to say, Jeff, thank you so much. Obviously, I have a million more questions for you, but this has been a really fantastic talk. Thank you so much for taking the time and giving us your insight. And it is really valuable to hear about, you know, not only the work that you're doing now and in the future, but that process for how it can be applied and brought to market, I think is something that people have been searching for a while. So really appreciate it. Thank you so much.
Yeah, thank you. It's been great.
Yes,
right,
absolutely.
Over to you.
Fascinating conversation we've had between you both, and then the chat going on. I know we could all keep this going if we could. Clearly, there are many approaches to take to looking to nature to solve problems, and there's value and excitement in creating these nature-inspired solutions. I'd like to reiterate for the group, you do not need to be a biologist or a scientist to tap into this wisdom. It's about finding the right tools and collaborators to further your research innovation. In the last few decades, technology has drastically increased our ability to capture information about newly discovered species and enhance the depth, the detail, and understanding of the species we already know. But as we can see from this engaging dialogue and the myriad of questions from you all, information is still dispersed. So our objective at the Institute is to capture and organize this knowledge and make it open and accessible to all of you to revolutionize industry as we know it. Like, imagine the problems we could overcome by finding solutions that nature has already created the blueprints for. How quickly could we further COVID-related research and collaborate with diverse thinkers like those at Karp Labs, and all of you who have mentioned some great projects that you're working on. Alex mentioned Ask Nature, obviously. This is the largest tool of its kind. It serves as a unique library of high quality, accessible scientific information that empowers civic science and direct connections between research and real world impact. There's currently no other function-based translation tool that shows how nature performs tasks that humans also seek to make, like filtering, adhesion, or ventilation strategies. And Alex also noted some new collections we're working on to organize these kinds of solutions. With your support, we will continue to grow this content so learners of all ages and speakers of all languages can readily understand these biological blueprints. The new Ask Nature platform will be rolled out in coming months, and new collections will focus on timely topics in science and innovation today. Our hope is to write about new species, especially those mentors that are endangered, and offer new innovations for the community. If you feel inspired to help us on this journey, please visit biomimicry.org. Every dollar can help us further this mission. And stay tuned for our meetup in July to be announced next week. We'll be diving into social and environmental justice and talk about how diversity and inclusion are essential to making the biomimicry movement flourish. Thank you all for being with us today. And a big special thank you to Jeffrey and Alexandra for taking us on this journey together.
Okay.
Thanks, everyone.
All right.
Bye.
Thanks, Lex.
Alex.
Bye.
Thanks, Lex.